Okay.
So I think there might be a few people drifting in, but thanks to those. I know we're a bit early in the semester for starting the series, but we had the opportunity, and I'd like to welcome Ryota Tamura from Keio University in Tokyo. So he's an academic neurosurgeon. He's got a broad interest in brain tumours, both research and clinically, including being the leading centre in Japan, probably Asia, for NF2 schwannomatosis and also glioma. So it's a very famous school, the Keio School for Neurosurgery. For anyone interested in neurosurgery, it was the home of Kawase, and so who's a very famous neurosurgeon, and Ryota is next in line coming down that lineage. So welcome, and thank you very much for sharing your talk with us, which is going to be about new biological treatments for brain tumours.
Okay, thank you very much. Hello everyone, my name is Ryota Tamura from Japan, and it is a great honor to be given this opportunity to present here. Thank you very much, Professor Omar, and thank you very much everyone. I have a slight hearing disturbance on my left side ear, so I may have to listen again a few times, sorry. Today I'm going to talk about the development of novel biological treatments for brain tumors. So let me introduce myself. I work at the Department of Neurosurgery, the Keio University School of Medicine in Japan, and my field of expertise is skull-based surgery, and awake craniotomy, and nasal endoscopic surgery, bypass surgery and cadaver dissection in clinical practice, and the stem cell-based gene therapy and immunotherapy and genome editing in basic research. So I focus on both the clinical and basic medicines. And there are many traditional cultures in Japan: kimono, sushi, samurai, and sumo. And this is a Japanese comic book, manga, called manga. It's one of our important traditional cultures: Dragon Ball and One Piece and Naruto. And my hometown is Tokyo, and Mount Fuji is considered a symbol of Japan. So please let me know when you come to Japan. I will show you around. And I work at the Keio University. The Keio University was founded by Yukichi Fukuzawa in 1858. And his face has been previously printed on the highest denomination of banknote. And the double pencil mark is a symbol of Keio University, meaning that the pencil is mightier than sword. And this is a university campus. And I belong to the Department of Neurosurgery, and I studied physiology as well under the Professor Hideyuki Okano. The past professors in neurosurgery, Takeshi Kawase, Kazunari Yoshida, and Masahiro Toda, specialize in the skull-based surgery. For example, the anterior transpetrosal approach, the Kawase's approach. His Takeshi Kawase was established in a hospital in 1985. This approach can provide direct access to the cerebropontine lesion with minimal brain interaction. So I also specialize in skull-based surgery. And every day I perform surgery and research for these diseases: schwannoma, meningioma, pitnet, chordoma, glioma, and other brain tumors. And today I focus on the translational research for schwannoma and glioma. So today's targeted diseases: refractory brain tumors, first NF2-related schwannomatosis, and second malignant glioma. And this is outline: the first VEGF and the VEGF receptor targeted therapy, the second precision medicine based on genetic analysis, and third research accelerating novel therapies. The schwannoma is originated from Schwann cell, the Schwann cell which forms myelin sheaths of an axon, supports neuronal function and regeneration. And schwannoma is one of the most common benign intracranial tumors. And 80 to 90% originate from the vestibular nerve, and among them, the 5% is observed as bilateral in NF2-related schwannomatosis. The genetic disease, and this is a typical pathology of schwannoma. And this is Schwann cell, and this is vestibular schwannoma. And what is the treatment strategy for schwannomas? Currently, the mainstays of management are observation, surgery, and radiosurgery. And the surgery with neuromonitoring remains the only curative treatment option for glowing schwannomas of all sizes. This is an example of vestibular schwannoma and a postoperative image, and this is MRI. And the stereotactic radiosurgery is considered as a widely accepted treatment option for small to medium-sized schwannomas, the gamma knife and the cyber knife. And for larger tumors, combined treatment strategies are mostly recommended. However, there is a refractory type of schwannoma, the NF2-related schwannomatosis. This is the autosomal dominantly inherited syndrome that predisposes the individuals to multiple brain tumors. And the diagnosis is based on the symptoms and the neuroimaging. The Manchester criteria is the most famous criteria in the world, and Professor Gareth Evans is the greatest researcher in the area of NF2. The NF2 patients develop schwannomas on the bilateral 8th cranial nerves and on other cranial nerves, spinal roots, or peripheral nerves. And the bilateral 8th cranial nerves cause deafness in most individuals. And NF2 patients often develop multiple meningiomas and ependymomas. And in Japan, the survival after diagnosis is 15 years, with an average age at death of 36 years. And a 10-year survival rate of 67%, and very rare disease. This is a representative case of male patient who has bilateral vestibular schwannomas, bilateral trigeminal schwannomas, meningioma, and multiple schwannomas. And the molecular function of NF2, SMARCB1, and LZTR1. The genetic testing may be considered medically necessary when the diagnosis is clinically suspected because of signs and symptoms of the disease, but the clinical diagnosis has not been made. And the NF2, SMARCB1, and LZTR1 are associated with schwannoma formation, and NF2 is the most important gene. The NF2 schwannoma is caused by a defect in the NF2 gene that normally produces myelin, located on chromosome 22, which regulates multiple proliferative signaling pathways. And SMARCB1 and LZTR1 are also tumor suppressor genes. And most NF2-associated schwannomas are managed conservatively in Japan because, for example, the vestibular schwannoma may involve facial nerve and cochlear nerve, possibly posing a significant risk of damage to the facial and the hearing function during surgery. This is my result at the Keio University Hospital, the vestibular nerve NF2 and non-NF2, the postoperative hearing preservation, and the postoperative facial nerve preservation rate. And the facial and hearing preservation rate were lower in NF2 patients than in non-NF2 patients. This is the intraoperative image of NF2 patients and the vestibular schwannomas and the lower cranial nerve schwannomas, multiple schwannomas. And the radiotherapy is a very good option, but is not advocated for multiple or large tumors. So how can we treat NF2 patients? Which is important, tumor growth or hearing loss? This is our data truncating very severe mutation type. The understanding NF2 genotype is important to predict natural cause of not only tumor growth but also hearing. This is hearing. And in natural cause, NF2 patients with an increased tumor volume were more likely to experience hearing deterioration. And a positive correlation was observed between hearing and tumor growth. So, regulation of tumor growth is important to preserve hearing in NF2 patients. And the molecular targeted therapies are important to regulate multiple tumors. This is NF2 gene-related signalings, and the many molecular targeted therapies are used for the NF2 patients. And this is a summary of recent clinical trial. The bevacizumab, brigatinib, and axitinib had efficient anti-tumor effects with fewer side effects for NF2 patients. And among them, the targeting VEGF and the VEGF-R is considered one of the best treatment strategies for NF2 schwannomatosis. And the VEGF-A, the main angiogenesis stimulator, suppresses anti-tumor immunity by inhibiting the maturation of dendritic cells and the induction of Tregs, TAMs, and MDSCs. And the VEGF receptor 1 and 2 are expressed on vascular endothelial cell, TAM, tumor-associated macrophages, and MDSCs, and regulatory T cells and tumor cells. In a previous study, the CD34 expressed vascular structures are evaluated in CNS tumors, the glioblastoma, NF2, and the chordoma, ependymoma, hemangiopericytoma. And NF2 schwannomatosis related to schwannomas have relatively large vessels, meaning the upregulated angiogenesis. And the NF2 gene mutation leads to the upregulation of the VEGF-A, and the VEGF receptor 1 and the VEGF receptor 2 are also upregulated in NF2 schwannomas and are expressed on not only vascular endothelial cells, but also tumor cells like this. So the anti-VEGF antibody bevacizumab was frequently used for NF2 patients from about 2009. The 40 to 50% of vestibular schwannomas partial response in tumor volume, 20% of the patients hearing improvement, duration of treatment 16 months median, and the complication 70%. So the bevacizumab is a good treatment option for NF2 patients, resulting in tumor control and hearing improvement. And the previous report showed the treatment effect of bevacizumab for sporadic schwannomas. But some aspects of bevacizumab treatment may be problematic. It requires frequent administration and has been associated with side effects, rebound phenomenon, and drug resistance. And it may be a little difficult for young patients to continue to receive an infusion every 3 or 4 weeks. And is there a treatment strategy that has fewer side effects and has a long-lasting therapeutic effect? We think the immunotherapy may be a treatment option for NF2 patients. The VEGF receptor 1 and 2 epitope peptide vaccine. The VEGF receptor 1 and 2 targeting cytotoxic T lymphocytes may kill a wide variety of cells associated with tumor growth, including tumor best cells, tumor cells, and immunosuppressive cells expressing VEGF receptor 1 and 2. In the memory-induced cytotoxic T lymphocytes after VEGF-R's vaccination may persistently suppress progression of multiple tumors, leading to the maintenance of ADL. So we have conducted a phase 1 and 2 clinical trial from 2016. The 16 patients with NF2 schwannomatosis were enrolled, and the HLA-restricted peptide was used. This is the schedule. The VEGF-R1 and R2 peptides were injected subcutaneously into infraaxillary and inguinal regions once a week for 4 weeks, and then once a month for 4 months, total of 8 injections over 5 months. And the total study duration was 1 year, including the follow-up periods after the vaccination, 5-month study treatment, and 7 months follow-up periods. This is a representative case. In this case, the previous bevacizumab treatment had been ineffective, like this, but percent change in tumor volume reduced after vaccination in this patient, trigeminal schwannoma. And the partial response was observed in the left vestibular schwannoma of case 2, 12 months after the first vaccination, which showed a minor response at 24 months after vaccination, and a stable disease at 36 months after the first vaccination, and now the stable disease at 6 years after the first vaccination. This is the 24 months before vaccination, and pre-vaccination in 12 months after the vaccination. And in case 3, the one cystic tumor showing rapidly grows became enlarged after the first vaccination, PD, in this case. And this is a very interesting case. In case 4, the partial response was observed in the left vestibular schwannoma 12 months after the first vaccination. However, progressive disease was observed in the right meningioma. The activation of the VEGF pathway is not the main driver of angiogenesis in meningioma. Meningioma showed few VEGF positive tumor cells. And this is a summary of tumor volume that we used the radiographic criteria published in other group. And this is stable, this is minor response and the partial response, and this marked vestibular schwannoma. And among the 16 patients, PR was observed in more than one schwannoma in 4 patients, and minor response was observed in 5, SD in 4, and PD in 3. And the percent change in tumor volume significantly decreased after vaccination. The mean change in a year period before vaccination is 32%, and after the vaccination, minus 5%. This is tumor blood volume, TBV, CT perfusion. In the 12 months TBV analysis, the 30% reduction was observed in 3 tumors, but an increase was observed in 14. This is a representative case. And the adverse events, 14 patients developed grade 1 or 2 local skin reactions at the injection site with indulation, redness, and swelling. And no patient developed injection site ulceration, and no delayed wound healing, gastrointestinal bleeding, coagulopathy, or nephropathy was observed. But grade 3 diverticulitis was observed in 1 patient, and grade 5 intracellular hemorrhage was observed in 1 patient. But the independent data monitoring committee concluded that these SAEs were not likely to be related to the vaccine. But grade 3 diverticulitis had previously occurred every few months in this patient before the vaccination, and dural AVF may have been the origin of hemorrhage. And hearing, this is word recognition score, and hearing on the beta side was evaluated in 13 years of 13 accessible patients before vaccination. Hearing as measured by change in WRS improved in 5 patients at 6 months and 2 patients at 12 months. The blue color improved, red color worse, and this is a representative case. And histopathological findings. The paired pre- and post-vaccination samples were obtained from 2 NF2 patients, and the number of FOXP3 positive cells were lower in tumors after the vaccination, and after the vaccination, greater number of diffuse cleaved caspase-3 positive cells were detected in tumors, suggesting that the vaccination induces apoptosis in tumor cells and tumor blood vessels. And these findings were also shown in paired pre- and post-vaccination samples obtained from 1 glioblastoma patient in another clinical trial. This is an image, V-mark, and the tumor blood vessel. So, the bevacizumab leads to better radiographic and hearing response rates compared with VEGF vaccination, and the adverse events were more frequent with bevacizumab compared with vaccination, and the VEGF vaccine tends to show a delayed clinical effect. And the long-term suppression of tumor growth was seen in a subset of patients in this clinical trial. So our treatment concept, bevacizumab can be used in patients with rapidly growing tumors needing urgent treatment, and the vaccination can be used in patients with slow-growing tumors needing long-term stability. So for the formulation of this vaccine, the randomized double-blind clinical trial of immunotherapy with HLA-2402 restricted peptide vaccine in NF2-related schwannomatosis. Immuno-NF2 study has begun in June this year. And we would like to establish the concept of immunotherapy for refractory benign brain tumors. But this treatment has some problems that must be overcome. Issue 1, sustained immunoreactivity of CTL. The tumor control rates gradually declined after 2 and 3 years after the vaccination. This is 2 years, 3 years. And sustained reactivity of CTL is important to induce long-term stability of tumors. Both VEGFR1 specific and R2 specific CTLs were induced in 11 per 16 patients at 1 year after the first vaccination. But CTL reactivity was not detected in some of our patients at 2 or 3 years. And most vaccine needs boosters to enhance immune memory. So in the immuno-NF2 study, a booster is designed to maintain immunity, total of 15 injections, and the necessity of the booster will be examined in the trial. And issue 2, meningioma. Among the 7 patients with a meningioma, minor response was observed in more than 1 meningioma in 2 patients. SD was observed in 3 and the PD in 2. So no patients showed PR. This is a representative case showing the SD 24 months before vaccine, pre-vaccine, and 12 months after the vaccine. And the percent change in tumor volume was not reduced after vaccination in 8 meningiomas. Mean change in a 1-year period before vaccination, 5%, and 1 year after vaccination, 48%. In the tumor blood volume analysis, one tumor exhibited a 30% decrease. However, the other 7 did not continue to decrease during the vaccination course. And the tumor microenvironment in meningioma may be associated with the effect of VEGF vaccine, and a few VEGF-R1 and R2 positive tumor cells were detected in meningiomas compared with schwannomas. And radiographic responses were limited in meningiomas, so meningiomas may require other treatment strategies. In issue 3, how to enhance the treatment effect. The combination therapy with VEGF peptide vaccine and anti-PD-1 antibody enhanced the treatment effect for glioma mouse model. This is our previous study, the control and the VEGF vaccine, anti-PD-1 antibody, and this is combination therapy. And this is the survival curve of the combination therapy. So this treatment strategy can be applied for NF2 patients. And issue 4, overcome the resistance to VEGF receptor targeted therapy, the mechanism of action of bevacizumab. That we have previously reported using human tumor specimens. This is malignant glioma, resected following bevacizumab therapy. That bevacizumab induces a normalization of vascular structure, this is pre and post, and the reduction of microvessel density, and improves tumor oxygenation. This is very dramatic change. And both FOXP3 regulatory T cells and CD163 positive tumor-associated macrophages, TAM, significantly decreased in the tumor specimens. This is malignant glioma, resected following bevacizumab therapy, so under the control of bevacizumab. And this is one of the mechanisms of resistance. The CA9 is one of the hypoxic markers, and in general, tumor microenvironment has 2 types of niche, hypoxic niche and perivascular niche. And by comparing tumors resected before treatment, following bevacizumab therapy, and at the time of recurrence, this is effective, this is refractory, tumor hypoxia was regained, while the microvessel density was still paradoxically lowered in recurrent tumors after bevacizumab therapy. So the hypoxia targeted treatment has a possibility to overcome the resistance of bevacizumab. And recently, the reports on the tumor microenvironment in schwannoma have included the expression of HIF1 alpha was detected in the nuclei of tumor cells. The expression of HIF1 alpha was observed in both NF2 and non-NF2 schwannomas, which was confirmed by immunohistochemistry and qPCR analysis, the blue color HIF1 alpha. And the number of CD163 positive cells and HIF1 alpha expression was significantly associated with the shorter progression-free survival in NF2 schwannomas. And many papers regarding tumor microenvironment of schwannomas was published by Professor Tumours. So we studied so much from these papers. In issue 5, future perspective, that despite surgery and radiation, the chordomas, other tumor, often progress and become refractory to further treatment. In chordomas, both VEGF-R1 and R2 were strongly expressed not only on vascular endothelial cells, but also on tumor cells. And the patient with chordoma, an example, the surgery 4 times and radiation therapy 2 times, and after the vaccination, the chordoma shrunk gradually. So the second topic, the precision medicine based on genetic analysis. Our multidisciplinary approach, the Department of Neurosurgery in our hospital collaborates with the Otology and Audiology Center and the Center for Medical Genomics and the Center for Cancer Genomics. And more than 100 NF patients have attended our hospital, and we have performed many translational research to improve the prognosis of NF patients. And genetic analysis is routinely performed in our hospital for patients with sporadic and NF schwannomas. And novel pathogenic genetic alteration is sometimes discovered for schwannomas. For example, non-vestibular schwannoma. The tumor DNA extracted from the tissue was routinely analyzed for all schwannomas using a 216 gene next generation sequencing panel, the copy number profiling, and in silico structural modeling. Germline NF2 testing was conducted following genetic counseling. And this is the first report describing the novel NF2 inflamed deletion in a jugular flamen schwannoma. No additional oncogenic alterations were identified, and germline NF2 testing was negative. And this is the importance of whole genome sequencing. In our department, the germline and somatic analysis were routinely performed, and large germline deletions of NF2 account for 2 to 5% of mutation-positive NF2 cases, and often go undetected via exon-based sequencing methods. Here we describe the clinical and molecular feature of a 50-year-old woman with a 0.66 megabase NF2 deletion. In this family, these principles reveal the paternally inherited this deletion by allelic inactivation through 2 additional somatic mutations, and a clear management pathway for relative risk relatives, illustrating how combined copy number analysis, sequencing, and pedigree evaluation will optimize care in hereditary NF2 schwannomatosis. And does schwannoma have different genetic background stratified by cranial nerve of origin? And this is the result of whole genome sequencing in 25 intracranial schwannomas, the vestibular schwannomas, and trigeminal schwannomas, and the lower cranial nerve schwannomas. And the sequence level NF2 alterations were detected in 18 tumors. And all vestibular schwannomas harbored NF2 variants, release only 30% schwannomas from other cranial nerves did. And other alterations except NF2 were rare. A germline LZTR1 mutation was found in 1 sporadic trigeminal schwannomas here. So genetic background may differ in the nerve of origin. In the previous study, the fusion gene was detected in 10% of cases with schwannomas, and this fusion activates the MEK-ERK signaling pathway, suggesting the potential efficacy of targeted therapy, such as the MEK inhibitor trametinib. However, some reports have noted the considerably lower frequency of fusion-positive cases, highlighting variability across studies. For example, a study from Taiwan reported a fusion-positive rate of only 1.3% in intracranial schwannomas, and Norwegian cohort showed an even lower rate of 0.8%. And this is a result using 237 intracranial schwannomas, and the comparison of this fusion gene positivity rates between cranial nerve origin is shown in this table. The fusion was identified in 12.7% of cases overall, with a particularly high frequency in trigeminal schwannomas, 25.9%. And there are no significant differences between NF2 and no-NF2 schwannomas. And the fusion-positive group showed a significantly higher proportion of recurrent cases with vestibular schwannoma, and there was a trend toward greater preoperative tumor growth speed rate in the fusion-positive group, and no significant associations were observed with histopathological Ki-67 index. And in vestibular schwannomas treated with nearly total removal and close total removal, the fusion-positive cases demonstrated significantly worse recurrence-free survival compared to fusion-negative cases. So fusion status may independently predict poor postoperative outcomes, even in cases achieving nearly total removal. And is precision medicine possible for the patients with refractory schwannomas? The prognostic biomarker is important for the precision medicine. And this is an example. This case was resistant to radiation therapy and showed strong edematous change, and this fusion gene positive. The prognostic biomarker is important, and our findings indicate that this fusion gene may define a molecular subset of intracranial schwannomas with more aggressive clinical behavior, so it can be used as a biomarker. And furthermore, the MEK inhibition could be considered as one potential therapeutic option for the fusion gene positive patients. And the neutrophil to lymphocyte ratio, NLR, may be another candidate of prognostic biomarker. This is our result. The preoperative NLR 2.21 before surgery was significantly associated with retreatment after sporadic schwannoma resection and cases with subtotal removal. So NLR may be a predictor of retreatment. So third topic, research accelerating novel therapies. This is our research team for schwannomas. Group 1, the animal model, 2, tumor microenvironment, 3, immunotherapy, the 4, genetic analysis and gene therapy, the 5, drug repositioning. So today my presentation will take 1 hour, so I would like to discuss these topics at another occasion. The second target disease, malignant glioma. This is outline the establishment of neural stem cell-based gene therapy and application for regenerative medicine. The malignant glioma is the most aggressive primary brain tumor with dismal prognosis characterized by diffuse infiltration into the normal brain parenchyma. In particular, glioblastoma has poor survival outcomes like this, despite comprehensive treatment strategies, so no significant improvement in OS, and the development of new therapeutic modalities is urgently needed. This is the intraoperative image, the tumor area and the normal area. The complete surgical resection of tumor is difficult because of diffuse infiltration into the normal brain parenchyma. And before describing our treatment strategy, there are 2 important backgrounds. The neural stem cell, the NSC and the mesenchymal stem cell, MSC, have tumor-trophic migratory capacity toward tumor cells. This is the image of migration. And the NSC and MSC would be the possible candidate as a cellular vehicle for the targeted delivery of therapeutic agents toward malignant glioma. And the NSC has ethical issues and is not easy to obtain from human embryos. The induced pluripotent stem cell, iPSC, was developed for human in 2007, offering multipotency and are able to differentiate into NSC. The iPSC has the possibility to overcome ethical and practical issues in the clinical application. And suicide gene. A transduced suicide gene converts a non-toxic prodrug into a toxic substance, thereby selectively damaging the transduced cells, the green-colored cell. And the famous suicide gene and prodrug system, HSV-TK suicide gene and the Ganschkrovil GCV prodrug, and the cytosine deaminase CD and 5-fluocytosine 5-FC prodrug. In the tumor cells not transduced with the suicide gene are killed together with suicide gene transduced cells, the yellow-colored cells. This is called as bystander killing effect. So our treatment strategy, the human iPSC-derived neural stem cells with the suicide gene system may cover the invasion area and have a considerably therapeutic potential for the treatment of glioblastoma. The tumor cell death efficacy and transplanted cell death, the safety. And this study provides answers to some important research questions associated with stem cell-based gene therapy. The first, tumor-trophic migratory capacity. The quantitative analysis of stem cell migration in the brain is not easy, so we frequently use the organotypic brain slice culture system. The brain tissues were obtained without perfusion and were sliced into 200 micrometer thick sections. And this brain section slice contains implanted stem cells and the glioma cells with different fluorescent proteins. And the quantitative analysis of tropism and migration in the brain was performed, culturing in the confocal microscope for 2 weeks, and cell trajectories are tracked. This is ex vivo analysis. This is an example of the slice culture and the visualization of diffuse infiltrative invasion of glioma stem cell. First, the primary culture was performed, and we successfully established the human glioma stem cell model, HG008 cell line. This cell invades toward the contralateral side through corpus callosum, very invasive characteristics. But the in vivo analysis needs 2 months to show this invasiveness. In contrast, the brain slice culture can quickly show the different invasive characteristics between glioma stem cells and the conventional glioma cell, U87. And we visualize the spatiotemporal dynamics of invasion of human glioma stem cells in an orthotopic xenograft mouse model using time-lapse imaging of slice cultures and 3-dimensional imaging of the optically cleared whole brains. This is an example of slice culture, the U87 cells in the mouse brain striatum forming the mass lesion. In contrast, this is HG008 glioma stem cell line in the striatum, shows very invasive characteristics, different from U87 cells. And the different invasive characteristics of glioma stem cells in brain was evaluated using this system: striatum, cortical area, subventricular zone, and corpus callosum. The glioma stem cells in the corpus callosum migrated more rapidly and are unidirectionally toward the contralateral side with pseudopod extension. And the brain size culture system ideally suited for temporal analysis. However, global visualization is difficult because brain size culture is only 200 micrometer thick. So for spatial analysis, tissue clearing methods enable 3-dimensional imaging of whole brain with cellular resolution. This is the whole brain clearing. The 3D imaging of the striatum revealed that these HG008 cells migrated in a helical pattern, twisting around the axon bundles. This is the example, the slice culture striatum, corpus callosum. The very straight, unidirectionally in base, and they extend the pseudopod. And this is the midline. And this is the brain clearing, the helical pattern around the axon bundles in the striatum. This is also HG008 cells. And the multiple implanted cells can be visualized using different fluorescent proteins. This is mouse striatum and the red color neural stem cells and green color glioma cells. Live imaging using slice culture. And the treatment effect can be also visualized using slice culture when treated with prodrug. The growth of NSC neural stem cells and glioma cells in the slice culture was dramatically inhibited. This is a bystander killing effect. The green color glioma cells and red color neural stem cells in the mouse striatum. After adding the prodrug, both cells were dramatically inhibited. So the different migratory characteristics between NSC and the mesenchymal stem cell, MSC in brain, was evaluated. The NSC iPSC derived and the fetal tissue derived. The MSC adipose tissue derived and the bone marrow derived we prepared. And the stem cells were implanted around the glioma cell forming a mass lesion in the slice like this. And the Rose diagram map shows the spatial distribution of NSCs and MSCs around the tumor, including both the number of cells in various directions and their distance toward the tumor center. And the iPSC NSC had higher tumor-trophic migratory capacity compared with MSC, and the iPSC NSC could cover the diffuse invasion area of HG008 cells, glioma stem cells. This is video. the green cell, the HG008 glioma stem cells, and red color cell neural stem cells derived from iPSCs. The covers invasion area. And the tumor-trophic migration signaling was evaluated. And in the absence of glioma cells, mesenchymal stem cells formed clumps. And the released iPSC NSCs exhibited diffuse engraftment. And the in silico ligand receptor pairing analysis based on RNA-seq datasets was used to identify signals underlying NSC-specific tumor tropism. And there are two important factors associated with tumor-trophic migratory capacity: the self-repulsive action associated with FB and FLIMB signaling, and the pathotropism associated with CXCL12 and CXCL4. So, the tumor-trophic migratory capacity, the neural stem cells for glioma. And issue 2, the therapeutic gene. The suicidal gene. We first select HSVTK, the most traditional suicidal gene, and the transduction of iPSCs is preferable for safe and stable supply in the clinical setting. The HSVTK was transduced into the iPSCs using the lentiviral vector. And in general, iPSCs differentiated into neural stem cells through embryonic body, EB. However, HSVTK iPSCs could not form EB, so stable constitutive expression of HSVTK was impossible because HSVTK is cytotoxic in iPSCs. And the nucleotide metabolism analysis suggested that the excessive accumulation of thymidine triphosphate caused by HSVTK expression resulted in an imbalance in the DNTP pool. This unbalanced state led to DNA synthesis inhibition and cell death in a process similar to a thymidine block, but more severe. So next we select the YCD-PRT, another suicidal gene. The YCD-PRT converts prodrug 5-FC, an antifungal medication, into the 5-FU anti-cancer drug. And the UPLT can increase prodrug sensitivity by almost 100 times. So the YCD-PRT was transduced into iPSCs using lentiviral vector, and transduced YCD-PRT was not cytotoxic, but was silenced during neuronal differentiation of human iPSCs. So established NSCs, neural stem cells, were all 5-FC prodrug resistant. And in general, the stable constitutive transgene expression by the viral vectors is difficult in human iPSCs. So the therapeutic gene YCD-PRT, but stable constitutive expression is needed. And therapeutic gene insertion into the specific genomic loci. The viral vectors integrate randomly into the host genome, which raises concerns about transgene silencing, insertional mutagenesis, and oncogene activation. So CRISPR-Cas9 was used for our experiment. And the YCD-PRT was inserted into the monoallelic GAPDH, M-GAPDH, and the biallelic GAPDH, B-GAPDH, and the monoallelic ACTB or AAVIS1 loci. And interestingly, the CD-NSC, the B-GAPDH, and the ACTB showed higher sensitivity to prodrug 5-FC than other CD-NSCs. But when YCD-PRT was inserted into biallelic GAPDH, the GAPDH expression was decreased, which was associated with a low neurosphere growth rate. Actually, the EDU incorporation into the neurospheres was significantly lower in CD-NSC B-GAPDH than in other CD-NSCs. And the gene expression associated with NSC was not changed among naive iPSC NSCs and CD-NSC ACTB and fetal tissue-derived NSC. So the most appropriate gene locus is ACTB that achieved stable constitutive transgene expression, and iPSC-derived therapeutic neural stem cell CD-NSC ACTB. And the human therapeutic CD-NSC was implanted into the HZ008 invasive glioma mouse model, and the luciferase-based signal intensity was decreased in the mice, treated mice, and this is a control group. And HE staining and venous-derived fluorescence demonstrated a reduced tumor volume in brains of treated mice, this compared with control. And the survival curve of treated mice was significantly prolonged compared with controls. And to evaluate T-cell mediated anti-tumor immune response, the mouse iPSC-derived CDNSC was implanted into the mouse glioma stem cell model. The Kaplan-Meier plots of treated mice indicate notable anti-tumor effects, and histological analysis showed the complete disappearance of glioma cells in the most mice. And CD8 positive T-cell numbers were significantly higher in brains of treated mice. And to evaluate safety, the green-colored CDNSC-ACTB were implanted into normal brains. The location 1, this is implanted site. The Ki67 positive cells were not present in brains after 5-FC administration, and the cleaved Caspar-3 positive cells were present in implanted striatum. And there were no significant differences in location 2 in Ki67 positive and nestin positive endogenous mouse neural and progenitor cell number in periventricular areas between 5-FC plus and minus brain sections. So the present study provided answers to some important research questions associated with stem cell-based gene therapy. Therefore, this research concept can disseminate biological and engineering advances. And the clinical trial using CDNSC-ACTB will start in a few years. And since iPSCs have multilineage differentiation potential, the genome-edited iPSCs contain the safeguard, a CCL gene, and therefore might be used as a platform to promote clinical studies using human iPSCs. So for example, the iPSC and SC expressing CDUPRT can be applied for safe regenerative medicine without a concern for tumorigenesis. So the last topic is application for regenerative medicine. The neuroprotective effects of genome-edited human iPS cell-derived neural stem cells on traumatic brain injury. Yes, okay. The few treatment options. And the iPSC has a potential to overcome the ethical and potential issues, and the can be applied for safe regenerative medicine. The CDNSCs, if transplanted into the contralateral side of the injury site, migrated toward the injury site. In the sequential illustration showing overall surgical procedures of the traumatic brain injury mouse model, cryo-injury method, showing incomplete paralysis, the rotarot and the beam test was used. And the beam walking test results were better for intervention group B compared with the control group, over the entire period. And the accelerating rotarot test performance also tended to improve in the intervention groups as tibia entered the chronic phase, with a significant difference at some time points. And the trend toward smaller area of Evans fluid leakage was noted in the intervention group on postoperative days 14 and 42. This is 14. And a large number of NSCs were observed migrating to the tibia region on day 63, and NSC has the strongest overlap with NUN, while only a few overlapped with GFAP. No NSCs positive for oligo-2 staining were observed. And the Ki67 positive cells persisted in the non-5FC treated group, whereas they were not detected in the 5FC treated group, suggesting that proliferative NSCs disappeared in the presence of 5FC. And to evaluate brain atrophy after TBI, the area of the left lateral ventricle was measured. The cross-sectional measured area of the left lateral ventricle was significantly more prominent in the control group compared with the intervention group. And the RNA-seq data demonstrated that some neurotrophic factor receptor and the neurotrophic factor genes were upregulated in NSCs compared with MSCs. So the suicidal gene transfer solved the concern from tumorigenesis of transplanted cells. Thus, the genome-edited iPSC-derived neural stem cells can be applied as a safe regenerative medicine. And today's targeted diseases, NF2-related schwannomatosis and malignant glioma. I studied these two topics and while in graduate school, and this stage, I will do my best. And I am very grateful to my supervisors Masahiro Toda and Hideyuki Okano for giving me the opportunity to do research. And I would like to thank all the juniors who are doing research with me. And I would like to collaborate with researchers in UK in the near future. I hope UK, Japan collaborative research. Thank you very much for your extension.